So let's just jump right in. We got 4 presentations to go. So our next speaker is Lisette Waits. And before the intermission, if you don't remember, it was a half hour ago, we did hear about the complex psychology of compromise and the politics of transnational cooperation. And an understanding of these topics, though, would have really come in handy now with as we go to the lowlands of Nepal, close to the Indian border, where distinguished professor and department head of Fish and Wildlife Sciences, Lisette Waits, participates in the Nepal Tiger Genome Project with a team of local researchers and American colleagues. Using tiger fecal samples, and this ties the first half to the second half if you didn't catch that connection. We're going to, Lisette and her team are using tiger fecal samples to develop a genetic database. And using that, it's helping them to determine where poaching is taking place and ultimately hoping to trace tiger body parts that have been sold on the black market back to the location where the animals were killed and potentially to the poachers themselves. So what an impact. You may not know, but there are fewer than 4,000 tigers alive in the world today. And the Nepal Tiger Genome Project is helping to understand how many tigers currently live in the jungles of southern Nepal and northeastern India, and how this endangered species can be protected in the future. So please welcome Lisette.
So thank you for that introduction, and thanks to everyone for coming. It's great to see so many students and so many friends here in the audience. So I do a lot of different international research, but I chose this project to talk about tonight because it's one of the most interesting and unique projects that I've had the opportunity to work on. And tigers are such awe-inspiring creatures, but they're globally endangered due to habitat loss and fragmentation. And there were once 100,000 tigers in the world, and as you heard, there are now less than 4,000. And that led to a global effort to save this species. So in the areas in orange on this slide are the areas of the historic range of the tiger. And now it's found only in the areas in yellow. And that's 7% of the original historic range. And for the talk today, I'm going to be narrowing in on the border between India and Nepal in the Terai Arc landscape. And this landscape includes 3 protected areas, or multiple protected areas, as well as human settlements and agricultural zones. And tigers dropped to less than 100 individuals after the Civil War, but the government has been working extensively to increase the population size for tigers. My research uses non-invasive genetic sampling, things that animals leave behind in the environment, to track tigers. And one of the reasons this has really increased over time, because, as you can see here, there's a lot of opportunity to collect fecal samples. My collaborators on this project are Marcella Kelly from Virginia Tech, Kanjan Thapa, who was a PhD student at Virginia Tech from Nepal, who now works for World Wildlife Fund Nepal, and Debesh Karmachara, who runs the Center for Molecular Dynamics Nepal. So the goals of our project were to develop methods to monitor tiger populations using camera trapping and fecal DNA analysis. We wanted to estimate genetic diversity, gene flow, and connectivity, and develop a database that could be used for forensic analyses, as well as develop wildlife genetics labs and expertise within Nepal. So this started by 3 Nepalese researchers coming here to the University of Idaho to learn genetic methods. Also in 2011, I went to Nepal and taught conservation genetics workshops and returned again in 2012 to do additional training. Now, during my first visit to Nepal, I also had the opportunity to go to Chitwan National Park for the launching of this project, which is a collaboration between the US and Nepalese governments. We trained the local park guards, there was an official lamp lighting ceremony, and this was clearly really important to both the US and the Nepalese governments. This was also my first opportunity to see tiger habitat. And we spent 4 days in Chitwan National Park setting camera traps and doing surveys. And amazingly, much of this work occurs from the back of an elephant. Also, to my great excitement, there's a huge amount of poop, as you can see here in the bottom slide. So there are just amazing things to see in Chitwan National Park. We have wild rhinos, wild elephants, giant termite mounds, grass much taller than my head, and peacocks flying through the air. So I didn't see a tiger, but I did see their tracks. In terms of our research methods, we had a 15 by 15 kilometer grid spread across this 24,000 square kilometer landscape, which included protected areas, community forest, and the corridors between them. We set up camera traps in each grid cell, and we collected fecal samples in each grid cell. At the same time, we needed to establish a genetic lab in Kathmandu. One of those things required having our own solar power because the power goes out a lot there and it's just normal. So we had to prepare for that. Also, we had US dignitaries like the ambassador visiting our lab and learning about our research in Nepal. In terms of our results, we collected 770 fecal samples across that range. 50% were tiger, 40% produced an individual ID, and we detected 78 tigers distributed across those protected areas. Their genetic diversity or heterozygosity levels are moderate, around 50 to 60%. We also wanted to know, are they moving across this landscape? Is there a lot of gene flow? If so, this figure here would show one color, but you can see three colors, which is showing there was a restriction of gene flow among these populations, but there's mixing of colors, which also tells us there was migration between populations. We detected 5 migrants, 1 from Bardia to Chitwan, 2 from Chitwan to Bardia, and 2 from Suklafanta to Bardia. So next I'm going to talk about our results from the poaching analyses. So we needed to develop a geo-reference database, and we were able to collect samples from 120 tigers. And these were the first genetic samples collected and analyzed in Nepal. We analyzed 15 putative tigers that had been poached. We did confirm they were tiger, and 9 were assigned to location. We found that Bardia was the hotspot for poaching, with 6 of these tigers, 2 from Suklafanta and 1 from Chitwan. So our methods for camera trapping are now used to track the population size of tigers in Nepal. And you can see things are looking good for tigers in Nepal. They're really increasing their population size, and this is because of extensive government efforts as well as citizens to stop this poaching that was occurring. So we accomplished our goals of building wildlife genetic capacity within Nepal, and the Center for Molecular Dynamics Nepal has gone on to study many different wildlife species and to develop genetic identification projects for native fish and plants. I've been able to continue my collaborations with the Center for Molecular Dynamics Nepal and with the World Wildlife Fund, and I now have a Nepalese student who focuses on red pandas. And with that, I would also like to acknowledge all the funders and contributors to this project. Thank you.
Thank you, Lisette. I'm on.